So now, this is always confusing the first time you bump into it, just because of the kind of the structure of the way human beings decided to describe the structure of proteins. And that is, we describe the structure of proteins, I hate to read word for word, due to four different aspects of them. And therefore we say that proteins can be organized at four different levels. That's another way we call it, okay? So all proteins consist of a chain of amino acids. Here, I know it's kind of small, right? But here's a specific protein. This is an enzyme that's in your saliva, for instance. It helps break up bacteria cells, so it's a quite important one. And every one of its amino acids from the first here to the last has been identified, and I know it's small to see, but these are little abbreviations for those amino acids. This description of very simply which amino acid is in which position, one after the other, is known as the primary structure of a protein. So every protein has to be organized at the primary level because every protein is a combination of amino acids. So the identification of those and the sequence, the order of them, that is what the primary structure of a protein is about. However, it is this primary structure that is then going to determine how this chain of amino acids folds up. And the way it folds up into a certain shape is going to be all about what its function is, determines the ultimate shape, which determines the ultimate function. So for instance, just using this one as an example, if this amino acid here attracted this amino acid here in a way that caused them to bond, then that would result in this little loop like this. And so it's the primary structure, the fact that these amino acids are where they are, which has led to that further level of organization that is about folding, which we refer to in the terms secondary and tertiary organization. Coming up next with this first. Here's the second place you're going to see hemoglobin, besides just a general example of a transport protein. Hemoglobin is a protein that red blood cells are full of, right? And their job is to grab oxygen, carry it around, let it off, but the little container they're in, so to speak, is a red blood cell. The structure of hemoglobin has everything to do with its ability, and this disease called sickle cell disease. Sickle cell disease is a classic example of how one tiny change in the structure of something can cause a big change in its function, in this case a deadly disease that we call sickle cell. So we have learned enough to identify the exact specific change. Hemoglobin, it turns out, as we're going to see in a little while, is made of a few different polypeptide chains, protein chains, and in one of them, in the sixth position from the end, this amino acid glutamine has been substituted for by this amino acid valine. Later we're going to see the reason for that is actually a change in the DNA in those cells. So this is the result of that, what we would call a mutation, right? And this one specific thing causes this molecule to fold up in a way to where these hemoglobins stick to each other and they stack on top of each other. And when they start to do that, they stretch a red blood cell out into this shape that reminded somebody of that farming tool called a sickle, right? A blade that you put at the end of a stick and you swing it and cut wheat, for instance, hence the name sickle cell disease. The problem here is, and that's all shown in this little video, the problem here is that when these red blood cells try to go through the smallest of your blood vessels, which are called capillaries, they don't fit too well, as you can imagine, because of their shape. They clog it, they stick, and now you get blood clots and clogging, and eventually this is lethal. So now to the next level of organization, the next description that we humans have for what we find proteins are all about. And that is, because of their primary structure, sometimes in certain places, a chain of amino acids will spiral up into what's called an alpha helix, a discovery that caused this person to win his first of 2 Nobel Prizes for science. Not many people have done that, right? And then we find only one other shape kind of like that, regular repeated, in this case angles that are folding, and this is called a beta pleated sheet. This little model that they have here shows 2 areas, one of which has an alpha helix shape, one of which has a beta pleated sheet shape. There's a few others here. Here's another helix, here's another helix, right? But because at least some parts of this protein are organized in a way that has regular and repeated patterns, these are 2 key words here, we say that this is organized at the secondary level. And this is very common that a protein will have these shapes. And last thing, these folds are caused by hydrogen bonds, which you can see here. Remember, they're symbolized by dotted lines. Whether it's the helix shape or the pleated shape, the folding is caused by hydrogen bonds, and the folding results in regular. What's that mean? Well, that means geometrically regular, like circular or angled, and repeated kinds of patterns. Now, the last little dot on the last one, and I can't go back on slides here because I don't know what will happen, says, did you notice that there are irregular folds in that model before? So pretend that you did. And so if we look at this much simpler model, right, here this purple ribbon represents a whole bunch of amino acids. And they didn't really specify any particular one, like putting them like beads on a chain. But for a few of them, they put their R group. Remember, the R group is the part of an amino acid that can be different. And so here's a bunch of R groups that, as you can see, have interacted with other R groups. How did they interact? Well, sometimes they're hydrogen bonds, like right here. Sometimes they're covalent bonds, like this covalent bond between 2 sulfurs that's called a disulfide bridge. And sometimes they're just positive negative interactions like ionic bonds. Point is, the irregular folds, which we humans have described as organization at the tertiary level, are due to many kinds of bonds, not just hydrogen bonds, and they are irregular, not regular and repeated. Virtually, not always, but virtually every protein has some kind of irregular folding. Okay, so here's a little small example of a place where this protein has some irregular folding. Now we get back to the star of the show, because here's where we see hemoglobin again, and we're going to see it in at least 3 more places this year as examples of different things. So when we humans say that proteins are organized by the quaternary level, it really has nothing to do with 4. Primary, secondary, tertiary, quaternary obviously are like 1, 2, 3, 4, but they don't have anything really to do with those numbers. This is just the language that was chosen long ago to describe these structures. What quaternary organization means, as you probably already read, is that for this one protein molecule, so this would represent one protein molecule, there are multiple polypeptide chains connected to form this one molecule. In this case, there's 1, 2, 3. And in the case of hemoglobin, there's 1, 2, 3, 4 separate polypeptide chains connected, bonded, covalently bonded somewhere along the way to make one molecule. Okay, so you can see here in collagen, right? This is collagen, the one your skin has a lot of. You can see here that they have illustrated for you the fact that this also clearly has secondary level organization, because they've shown there that this has alpha helical structure. Well, for whatever reason, they didn't show that over here, because these also are strings of amino acid that have helixed up. But unlike over here, where you don't have any foldings that you would call irregular, here you have a lot of foldings that you would call irregular. And those are caused by R group interactions. And so, hemoglobin, why it's such a classically used example, is an example of a protein that's organized at all 4 levels. These polypeptide chains all have a specific order of amino acids. That's their primary structure. They then helix up due to hydrogen bonding into alpha helixes, secondary organization. They then take some irregular twists and turns due to R group interaction, tertiary organization. And then, since this one hemoglobin molecule has 4 separate ones stuck together, it is organized at the quaternary level.